Okay, so our next speaker is Dr. Aman Chauhan. He is a medical oncologist specializing in thoracic and neuroendocrine tumors at the University of Miami Sylvester Cancer Center. He did his medicine pediatric residency here in New Orleans at LSU. This is where we first met, and I tried to recruit him to stay on for his fellowship, but he had a girlfriend that he wanted to get closer to, so he ended up moving away and did his fellowship at University of Kentucky, where he stayed on staff for several years and built a name for himself within Kentucky, within the United States, and around the world as a leader in thoracic oncology and neuroendocrine oncology. He most recently has moved to University of Miami, and he's been there 6 months. A year, oh gosh, a year already. Yes, and is really brought the experience of treating these patients to South Florida, and so we're happy to have you here, and he's going to discuss what's new in small cell lung cancer. Dr. Chauhan.
Thank you, Dr. Ramirez, for the kind introduction. First of all, wonderful meeting, CAGLA. Congratulations. Especially on a Saturday late today. We have a packed house, so congratulations. And thank you, Dr. Ramirez, the section chair, for inviting me here. It's difficult to follow a surgeon's talk, so because they have beautiful pictures and they do magic with their procedures, but I will try my best, okay? Today, we're going to be talking about another such really poor prognosis cancer called small cell lung cancer. Before we proceed, these are my disclosures. So why do we have to worry about small cell lung cancer? Unlike mesothelioma, it's not a rare cancer. About 14 to 18% of all lung cancers are small cell lung cancers based on the location and the geography. If you're on West Coast or East Coast, because of the decline in smoking prevalence, we don't see that much of small cell lung cancer. But when I was in Kentucky and the whole tobacco belt and a lot in South Florida, this is definitely on the spectrum of 14 to 18% of all lung cancers. What is even more perplexing is the outcome of these cancers. While we have made huge strides in how we manage lung cancers, and now routinely our patients survive 5 years and longer with targeted therapies and immunotherapies, we have hardly made any dent in overall survival of small cell lung cancer in the past 5 decades. There is some ray of hope, and we will be discussing today, and I think the future is bright, though. Back when I was born, or maybe even before that, we used to treat patients with the regimen, very toxic regimen called CAV. And in the 1980s, we adopted a platinum doublet-based regimen because the efficacy was similar, but safety was much higher. And guess what? Since 1980s and 2024, we still are using the same platinum doublet for the frontline management. The first breakthrough happened a few years back with a study called IMPAR 133, where we added immunotherapy to our standard of care frontline chemotherapy. So we had 2 sets of groups. Half of the patient got standard of care platinum doublet, carboplatin etoposide, and other half received chemo plus atezolizumab. And what we see here is patient who received immunotherapy, they lived longer. Now, it is a marginal improvement, about 2 months, but this was a significant step up after years of drug development, and finally something was positive. And it wasn't just a lone phenomena. Similar other immune checkpoint inhibitors, for example, durvalumab, was tested in a very similar design study called Caspian study, and again validated patients who got chemo-immuno versus just chemo, they had about 2 months survival advantage. These studies paved the way for changing of the frontline management or standard of care management of small cell lung cancer for extensive stage. And now chemoimmunotherapies are routinely used standard care regimen. Okay, so that was good. But what about second-line therapies? Now, frontline treatment with platinum doublet, the reason why it is still one of our backbones of the treatment, it works really well. If I give platinum doublet to about 10 patients, 8 will respond. That's how fantastic the responses are. The problem is the duration of the response. Most patients would have relapse by 4 to 6 months. And that's where we are focusing on how to improve the durability of response. And immunotherapy sort of did that a little bit for us. Lurbinectedin is the nucleotide in the block. It's been there now for a couple of years. It was tested in a small phase 2 study where we did see some impressive response rate. For the unworse, response rates mean the capability of a treatment to shrink the tumor in a common parlance. So for historical control, when we are treating with small cell lung cancer patient, second, third, fourth line, the response rates are typically 10% or under. This is a very aggressive disease once it starts failing platinum doublet. So when we saw about 30% response rate, this got everybody excited. And a very small study led to an approval for lurbinectedin. I think there is definitely a role for lurbinectedin. I'm a very heavy user because we don't have other good alternatives. But we also think we need to continue to make these incremental improvements for overall patients. Now, this is a laundry list of all the drugs that currently NCCN guidelines mention that we can potentially use. A lot of them don't have very strong randomized data, but when you're dealing with a recalcitrant cancer with 5-year survival under 10%, we are desperate. So we try irinotecan, we try, and immunotherapy, nivolumab, Keytruda were approved, but now there've been some, pullback on that because immune checkpoint inhibitor now made it to the frontline therapy, paclitaxel, docetaxel, even single-agent Temodar, which is a pill form of chemo that we commonly use for brain tumors, also have some activity in small cell lung cancer. All in all, it's good to see many options, but they're not that great. It's not like breast cancer that we can try 5th, 6th, 7th line treatment and they work. The response rate with these 5th, 6th, 7th line treatments are under 10% and retain a lot of toxicity. So why all such doom and gloom and why am I even here? Well, the times are changing and we might have some very, very promising agents on the horizon. Let's start with the first of its kind. So there are, I think, at least 3 companies who are testing this class of drug called DLL3 targeted BiTE antibodies. I think Amgen took a lead and their studies are very advanced, followed by BI. And there's a small biotech, I think Rocket, which is recently acquired. And I'm sure there'll be many more companies who will be investigating this class of drug. So what is it? These high-grade neuroendocrine cancers, like small cell lung cancers, they express this protein on their surface called DLL3. Now, if there is a target and you have a drug that binds to that, and then you can engage your immune system to fight that cancer cell by attaching to that, that's the basic concept. So this BiTE antibody, which stands for bispecific T-cell engager, think of this antibody with 2 arms, bi. One of the arms is attaching to the DLL3, which is present in the tumor, and the other arm is attaching to T-cell. That's what's a T-cell engager. What is a T-cell? That's your own immune system. That's a circulating killer cell. So if you inject this drug and it latches onto the cancer and it's tried to kind of then attract the T-cell and bring it closer to the cancer, and then your own immune system does the rest. So it's a form of immunotherapy where your own immune system is engaged to kill the cancer. So before we look into the efficacy data for some of these drugs, are they safe? Well, there is a small concern for autoimmune side effects. One of those which are very much linked and studied with this class of BiTE class of drugs called CRS, cytokine release syndrome. Imagine we are trying to engage and rev up the immune system, try to fight the cancer. But if we do it a little bit more, you can actually make it worse. And there's a cytokine storm and all these chemicals which are supposed to not just kill the cancer, starts affecting your normal organs. Fortunately, most patients who had this CRS syndrome were grade 1 or grade 2. That means fevers or chills, and that too for the first day most of the time. And this side effect usually dissipates with subsequent cycles. And that's why most of this trials who study this class of drug did a step-up dosing. They gave a very tiny dose, then a week later they had a little bit bigger dose, and then a bigger dose. And by the time second or third cycle came, the body was kind of used to it, and we didn't even see that high-grade CRS syndrome. The other unique side effect that we as an oncologist are now learning is called ICANS. This is, again, one of those autoimmune side effects causes encephalopathy. We don't want this immune system to go haywire, but if it does, sometimes it can affect your CNS tissues. Again, serious side effects, but fortunately, very rare. Now look at the benefit that we've been seeing from some of these early phase studies. And this is sort of very true for not the BI drug, but the Amgen drug and the others in the development. The response rates are in 30 to 40% range. Remember, guys, we discussed the traditional refractory small cell patients, the response is under 10%. And when we start seeing 30 to 40% patients are showing response, very refractory patient, that gets all of us excited. And which is more important is the durability of the response. That means they just don't shrink the tumor for a month or two. The patients often benefit for a long duration of time. Now, let me put a little human touch to this. So when I was in Kentucky and I was involved, I won't name names because some of these data are still not published. But I was involved in some of these early phase first-in-human studies with this class of drug. And this patient found us through clinicaltrials.gov. I said, Doc, my dad has small cell lung cancer, progressed on 6 lines of therapy, but still very fit. And I'm pregnant. I want my dad to live for 4 months so that he can see the grandkid. And, luckily for us, this study had just freshly opened, and we enrolled the patient. 2 years later, complete response. The patient was gallivanting in California. Now I'm in Miami, so I'm hoping the patient's still doing good. But that's the kind of profound responses that when you see kind of really is paradigm shifting and encourages you to look hard. And so we are all very excited. This is a graph that shows durability of responses. So these arrows are the number of months the patients are staying on these drugs, and very highly refractory patients on multiple lines of therapy. So in summary, I won't go with too much of it, but this class of drug, DLL3 BiTE targeting, regardless of what company or what agent you're going across the spectrum, the data looks very exciting. Be it Amgen data, you can see it from ASCOs and AACRs from last year, or BI data from the World Lung Conference. It looks very, very promising. All right, besides the DLL3 story, what are some of the other potential breakthroughs that can be seen in the near future? So last year, World Lung Conference was in Singapore, and one of the Chinese study tested their homegrown drug, which is an immune checkpoint inhibitor, and they added a VEGF TKI to this regimen. Now, there is plenty of data, preclinical data, which suggest that when you add immune checkpoint inhibitor to a VEGF drug, it really sensitizes, make these cold tumors hot, okay? So that was the rationale, and they had this large randomized study with 3 arms where they added standard of care chemotherapy plus immuno, standard of care chemotherapy plus immuno plus VEGF TKI, and just standard of care chemotherapy. Some of the data were available, and look at these Kaplan-Meier curves. It is very exciting. So the median PFS, when you added these 2 agents to standard of care chemotherapy, the median PFS increased from 4.2 months to 6.9. This is statistically significant, so this is not a fluke. This is real. But even more importantly, look at the overall survival change from 11.9 months to 19.3 months. Again, statistically significant. Now, I have to caution you, there have been studies that have been done in East that have not panned out in West. Could be difference in how patients metabolize the drugs, the pharmacogenomics of it. But this is very solid data, and I think time is ripe for us to have these kind of studies here in the US population because this does look very profound. The other data that we learned from recent World Lung Conference in last fall is we discussed the IMPAR 133. The addition of immunotherapy led to 2-month overall survival benefit, which paved the way for this being a standard of care in the US. But we started off with saying that 5-year survival is under about 7 to 10%. Has there been change in the era of immunotherapy in the long-term survival with small cell lung cancer? And this study, the expansion cohort, gave us some of those clues. And we did see some incremental increase. For now, this 5-year survival from 7%, we can say it's up to 12%. So for those who benefit from it, it's a significant boon. We now have to focus on the other 78% or 80% patients, 90% patients to how to get them into remission long term. There are some other potential concepts that are being tested and looking very promising. One of them is using a drug called PARP inhibitor. You've probably heard of these drugs used in ovarian cancers and some other cancers. But there's very solid preclinical data. When you add PARP inhibitor to checkpoint inhibitor, it does sensitize these cancers to respond better. So we ourselves in Kentucky did a phase 2 study using a PARP inhibitor, rucaparib, and added it to nivolumab as a maintenance agent after frontline chemotherapy. And from our single center experience where we treated close to 30 patients, 90% of patients are alive at 1 year and about 50% at 2 years. Of course, one of the key factors here was we were selecting platinum-sensitive patients because historically we know that PARP inhibitor was best in platinum-sensitive population. But that really gives us a proof of sort of a concept here that if you can bioselect patient for platinum sensitivity and add PARP inhibition in frontline maintenance, there might be some benefit in doing that. Dr. Nagla Karim, one of our colleagues, also have a very similar concept and did a much bigger study, a cooperative group randomized study. And they added PARP inhibitor to frontline, talazoparib to frontline immunotherapy, but they picked SLFN11 as a biomarker because based on their preclinical data, they feel SLFN11 positive patients are more sensitive. And they were able to show a median PFS benefit, although marginal, from 2.8 months to 4.2 months. But I'm very curious to see how the overall survival data pans out maybe in a year or two. I would finish off my talk with one of the hats that I wear in Miami as I co-lead our theranostics or radiopharmaceutical drug development program. And there's a lot of activity when it comes to theranostics in solid tumor period. You've probably heard, BMS acquiring Raise Bio and, Lilly investing $4 billion. So this whole radiopharmaceutical field kind of is expected to grow to about $16 billion in just next 5 to 7 years. So what's the role of theranostics in small cell lung cancer? Theranostics is a concept where we have a target on the cancer and we develop a drug that likes to attach to that target. And then we can attach a radionuclide to that drug. So when we inject that drug, we can not only see the targets in real time with the help of a PET scan, and if patient does have those targets in abundance, change that radionuclides to a heavy-duty radionuclide, not to diagnose, but to now treat it, to radiate and cause DNA damage. And we have 2 successful drugs. Lutathera, Dr. Ramirez have probably taken hundreds of patients with Lutathera, and NETs patient works very well. And Pluvicto, the recent approval in prostate cancer, again, another game-changer drug. But this is just the scratch of the surface. These class of drugs will spill into prostate cancer, pancreatic cancer, breast cancers, and there are a lot of really exciting targets. But for today, we will focus on small cell lung cancer. So we did a small study. We wanted to investigate somatostatin receptors in different solid tumors, but more specifically in high-grade neuroendocrine cancers like small cell lung cancers. Because we have a drug like Lutathera, which is radiopharmaceutical and does a great job by radiating somatostatin receptor positive tumors. So what we found, about 60% of patients' samples were positive for some degree of somatostatin receptors, and about 30% were truly positive, were hot on IHC. And you really want some significant uptake for the drugs to work. There has been already some human studies that using SSTR targeting in small cell lung cancers. This small study investigating 4 rounds of PRRT, and you can see significant shrinkage in the tumor burden before and after. Why does this happen? Small cell lung cancer is very, very radio-sensitive tumors. Be it radionuclide-based radiation or external beam radiation, it dies, rather briskly with radiation exposure. There's also a small study which was done in Georgetown testing PRRT plus immune checkpoint inhibitor nivolumab in small cell lung cancer, and they were able to express that it is safe to do. Although the efficacy data was limited, we don't have that in published. But this kind of gives us some legs to stand on, that we have a great drug which targets SSTR, and maybe we should look into non-NETs indications like small cell lung cancer, which are area of critical unmet need. And without kind of going into further details, you will see in the near future a couple of clinical trials investigating both beta and alpha PRRT agents in this small cell lung cancer space. So it's just getting exciting. As I mentioned before, there are a lot more targets and a lot more indications that might be investigated in the near future. One of those targets, fibroblast activating protein, is expressed in a lot of different cancers. Look at the gallium scans for FAPI, and it expresses all those black dots are tumors, so breast cancer, pancreatic cancer, colorectal cancers. Can we leverage FAP for small cell lung cancer? There is reason to believe so. This is very early in the drug development, so I would hope the companies who are thinking about developing these drugs also consider small cell lung cancer and other similar cancers who have potential or are critical unmet needs. Other such target that I'm very curious about is called integrin. You can find integrin in a lot of solid tumors, and there's preclinical data in various solid tumors, and you can radio-label it and then later treat it with theranostics. We believe that it is also expressed in small cell lung cancer. But again, now we are going from mainstream DLL3 to really pipeline preclinical data. The goal here was to showcase that we are making progress, and there are a lot of other agents in the pipeline that hopefully will transform how we manage. And I would like to conclude by saying DLL3, great story with the BiTE targeting, but why can't we target DLL3 through radiopharmaceuticals? And I think there are going to be studies in very near future that you will see in first in human settings that will be testing radiopharmaceutical antibodies linked to DLL3. And because this is something that is expressed heavily, be it thoracic small cell or extrathoracic high-grade neuroendocrine cancer. So to summarize, most small cell lung cancer patients at time of diagnosis unfortunately have advanced disease, so they are not curative candidates. And frontline treatment has now acquired small immunotherapy as a part of chemotherapy combination with some improvement in outcomes. We are still in hunt for better second, third line treatments. DLL3 BiTE probably is, if I have to bet on I'm a Kentucky, I was in Kentucky a little bit, so if I'm a betting man on horse, DLL3 seems to be the drug that has the most promise out of the lot for now. But there are so many other promising agents, including radiopharmaceuticals. Last 2 slides, just want to show our audience, neuroendocrine cancer is not one disease. It's a spectrum of disease. So small cell lung cancer falls on the most aggressive end of the spectrum called neuroendocrine carcinomas. And we need multidisciplinary programs. Dr. Ramirez leads one such at Vanderbilt, and we have it in University of Miami as well, so that we can create an infrastructure and provide latest and greatest clinical trials to our patients. Because if you segregate these cancers in thoracic and GI and other groups, then it becomes very hard to kind of open several clinical trials and promising innovative treatments for them. With that, I'd like to happy to take any questions.